And the recording has started. So now, more to you.
Awesome. Right. Thank you everyone for having me. So I'm glad it's going to be you're going to translate into Russian because my Russian's terrible. So we're going to talk about power analysis. So hopefully, as an audience interested in this thing, it's not going to do too much on convincing you why it's important. Although I do touch on it for some important points later, but it's more about some of the practical issues, some of decision making, and then for the middle parts, we're going to hopefully get you doing some of the power analysis just to demonstrate that you've done it. You can sort of follow along. So hopefully, if we go to the next one, if you'd like the slides, some of the screenshots later might be a bit difficult to see. So you can download the slides, and this link will take you directly there. It might be slightly different to for the one Ilya shared the other day. I've just made a specific one, and this will take you to the direct slides. And hopefully you will either GPower or Jamovi downloaded. So the reason there's two options is so I use a Mac myself, and if you have the most recent download, it doesn't support 32-bit software, so GPower doesn't work on my Mac anymore. So the things we're going to demonstrate, you can use it whatever computer you're using. It will just whichever one you can download. So I'm mainly going to focus on the concept of statistical power on relatively simple design. So we've got two groups, two conditions, slightly more complicated designs in terms of factorial experiments. So mainly going to be focusing on power for experimental design or quasi-experimental, just separating people into groups or conditions. What we're not going to be covering, just because I wasn't 100% sure what level people was coming into it at, so I'm going to focus more on the concepts and being able to apply the power analysis procedure to something relatively straightforward, very accessible. And then the idea is that hopefully you can take these skills and apply them to more complicated models. So we're not going to talk about it specifically today. So things like regression, mixed effects models, but one of the things I've put in the folder that I've linked to is an annotated reading list. So if on the back of this, you need to apply it to something slightly different, I've put a collection of resources that I have that hopefully you can apply what you learned today to something a bit more specific to your needs. And sort of following from this, I wasn't sure whether people have much experience in things like R or Python. So that's going to give you the most flexible approach to power analysis. But if you don't have experience in it already, it again provides quite a high bar. So I was focused on more of the accessible side. And then two of the other things that I've included in the annotated bibliography is things about Bayesian statistics, sequential analysis. That's something you're aware of. And then we can, for the Bayesian stats, power analysis works a little bit differently, has slightly different focus. And then sequential analysis, it's when you're not 100% sure about the effect sizes you're interested in, so it can be a little bit more flexible. So I've provided some additional resources for that as well. So to ground things in for the concepts, so we're assuming here that we're using things like p-values, so null hypothesis significance testing. It follows what's called a frequentist concept of probability. So probability is applied to procedures rather than individual events. And the idea is that it can help you to control your error rates. So it's not the strength of evidence, it's just we want to set boundaries for the amount of type 1, type 2 errors we expect. So depending on whether we're rejecting the null hypothesis or not, we're creating these boundaries, and this is where it allows us to get these probabilities in. So for example, applied to power, it's usually we're aiming for about 80% power. So the idea is if you was to repeat a procedure over and over again, you would expect 80% of those results to be statistically significant, and 20 of them you wouldn't expect. So it's trying to create these boundaries, and through creating them, this is where we can work out these probabilities, apply it to power analysis. So what we're going to be focusing on for power is, you have to imagine, so if you have an effect of a certain size, how likely am I to detect it? So that's where the probability is coming in. There are slightly different approaches to power analysis. So the two really informative ones are what's called a priori. So when you do this before a study, so it's most useful in the planning stages, so before you start collecting data, which is for a given effect size, which is going to be the main focus today. How many participants would I need to detect it with X power? So typically we use 80, 90%, those kind of regions. But then we can also look at what's called sensitivity power analysis. This is most useful after you've conducted a study. So rather than stating an effect size and working out how many participants you would need to detect it, it's now that you know the sample size because you've conducted the study, you have your participants, it's now the question flips, and it's what effect size would I be able to detect with X amount of power? One of the things that you see in different places, but it's really not very useful, is what's called post hoc power. So things like SPSS will give it you, and it's attractive because it makes it sound like, oh, based on the result I found, how much power did I have to detect it? But it's not really consistent with the theory of probability we're working with, because it's applying essentially probability to an individual event where it applies to a procedure. So post hoc power is essentially your p-value just expressed differently. Essentially, in any one study, if you've got a significant result, you had enough power. If you didn't have a significant result, you didn't have enough power. So it's not really informative. So we're not really going to focus on it today. So one of the things I would like you to do, depending on whether you've made your way through the slides over the weekend or not, is one informative exercise is trying to calibrate our idea of what power or what, like, a good sense of power. So one of the studies that's in the reference list, they did a really cool experiment where they told people, right, assuming this effect size, how many participants would you need? Then the flip side, if you know this many participants, what effect sizes could you detect? So if you could, in the comments, if you want to detect a medium effect, so currency of 0.5, you have two groups. How many participants do you think you would need in each group to detect this effect with 80% power? So you can have a complete stab in the dark. How many participants do you think you would need? 26. 52, 36. 5. So again, a bit of a range. So we've gone from 5 to 36. So it'd be really helpful to read that paper and see what they found in their study. So everyone was an underestimate. So even with 52, it's actually 64 per group. So for 80% power, two-tailed, be able to do this yourself a little bit later on. You would need 64 people in each group, 128 people overall, to detect that with 80% power. So it's the same as what Bakker and colleagues found, where people consistently underestimate how many people they would need to reliably detect a given effect. And when they flipped it around and said, how many people would you need to detect this effect? People would be, sorry, explained that wrong. Given a known sample size, what effect size could you detect with X amount of power? They consistently overestimated it. So they thought they could detect effects far smaller than what they could. So coming on to the, following on to the like the frequentist idea of where power comes from. There's a collection of visualizations. A guy called Christopher Magnuson has a really good collection of visualizations to do with different statistical concepts. So this is where the idea of power comes in. So we're assuming there is an effect. So this is where the idea of these type 2 errors come in. So how likely are you to sort of fail to reject the null if there is a true effect? So if we assume that there's a medium effect, like we just used in the previous exercise, the idea is that if you repeated this test over and over again, and you counted the number of p-values above and below 0.05, this is where the probability comes in. So if we have enough power, or at least adequate power, some people argue that 80% is still low. What it's saying is that there are 80% of those p-values would be between just above 0 and 0.05. And then 20% of them would be between 0.05 and 1. So the probability comes from these sort of sequences of events. So how many times it falls below 0.05, how many times it falls above. And then if we have low statistical power, this gets adapted slightly. So rather than 80 odd people per group for a medium effect, we're now looking at 25 per group. So we're still assuming that there is an effect, but the distribution slightly changes. So rather than there be 80% between 0 and 0.05, we now have 33% of them. And then 67% are between 0.05 and 1. So this is where this idea of low power comes in. If we are trying to detect a given effect size and we have a too small a sample, most of the time you're just not going to find anything. And then the only things that you are going to find are massive, just by chance. But most of the time you're just not going to find anything at all, because that's how, if you were to repeat it over this long rung frequency, you would just expect very few of them to be below 0.05 and reject the null when it should be. So I said I wasn't going to focus too much on the actual convincing you because you're here, but hopefully the last bit will demonstrate that it's really trying to prevent wasting your own time. So both your time conducting a study that's not going to give you very informative results, and also your participants' time. So you can almost bring in an ethical argument if you're asking people to give up their time to contribute to research that's never going to do much. You could argue that's an ethical issue about whether you're wasting their time or not. But then the concrete aspects of it is sort of alluded to on that last slide, where when you have underpowered research, it's like this weird paradox where you can only detect large effects. So when you combine this with publication bias, so if you only publish the things that are statistically significant, you get this like warped sense of reality where the only things you ever observe are huge effects. And when research tries to replicate this, you do a power analysis on huge effects and you convince yourself that you're going to find something with X amount of power. You're going to underestimate the sample sizes you need. And to sort of demonstrate this, I talked about it on the distribution of p-values, but power isn't a single number. So one of the, it's like the second most important concept is it exists along a curve. So if you use Jamovi to do power analysis, you'll get this graph. You can also get G power to calculate a power curve as well. So you can see that if for an effect size of 0.5, you would need 64 participants. So around the yellow area, so it's just where it's turning green, that would be where 80% power is. And we can see for 80% power to detect an effect size of 0.5, you need 64 participants. But then if you follow this curve along, as the sample size gets smaller, if you look on the x-axis, say if you had 10 participants, the only effect that you're detecting with 80% power is around 1.4. So as the sample size gets smaller, the effect size that you can reliably detect gets bigger and bigger. So this explains this weird paradox where if you're constantly doing results with small samples, if you repeat it over and over, most of the time you're going to find nothing. So the effects are going to be what they are, and you're not going to find anything. But very occasionally, just because of sampling error, you're using small samples, you will get a huge effect size. Just by chance, you will observe a huge difference between your groups, and that will be statistically significant. And because that is the only effect size you can reliably detect, you get one study, say out of 20, that are going to have a huge effect. And because of publication bias, that tends to be the one that gets published. So all you would see is the one study that found this huge effect, and the 19 studies that found reasonable effects but were just non-significant, they get hidden, which is the kind of core issue around why power analysis is so important. So to focus on some of the practical issues, the single hardest part of power analysis is actually what effect size you're interested in. So we've used 0.5 for the examples, but then the problem is you don't know what the effect size is. So this is the single biggest problem here. So if you knew the exact effect size, you wouldn't need to conduct the study in the first place because you would know the answer. So we're trying to make an informed guess about what effects we're interested in, or the very least, which would you not want to miss. So this brings in this idea of the smallest effect size of interest. So rather than saying we expect the effect to be precisely this value, the idea is more to go below that and go, which wouldn't we want to miss? Because we don't know the true effect size, we're trying to provide like this informed decision to estimate what we would be looking for. So best case scenario, which you will need for the more complicated power analyses, is like an informed idea about what values you expect based on previous research. So for example, the means and standard deviations, if you're looking at within subjects designs, the correlations between them. So you need quite a lot of information to have a really fully informed power analysis. But then the next best thing you can have is subject-specific effect size distributions. So everyone knows Cohen's guidelines, so 0.2, 5.8. So they're used all over, but there is essentially Jacob Cohen guessing. They sometimes don't apply very well to different areas. So everyone uses them as rules of thumb everywhere. I kind of use 0.5 as my own. So going from the examples, usually because of this next example. So there's this huge study. It's quite old now, so it's probably a little bit of an overestimate, but they combined like 20,000 studies and looked at the average effect size across social psychology. And it was around D equals 0.43. So I use this as my personal like barometer of what effect size would I expect. If I've got absolutely nothing else to go on, that is probably where I'm going to go. So it's like this, just smaller than Cohen's medium. And if I've got nothing else to go on, that is potentially potentially the next best thing. But then there is this whole series of other studies that for specific sub areas of psychology or specific topics, they've tried to look at these distributions. So for example, in pre-registered studies, that you have far smaller sample sizes, effect sizes, sorry, which is probably a byproduct of they tend to use far larger sample sizes so you can find much smaller effects. So here it goes to 0.08, 0.3, 0.9, so quite a sparse distribution. You've got specific to neuroscience research, these tend to be a lot higher, so from 0.64 to 1.46. Although in this study as well, they break it down to significant and non-significant, and the non-significant results are pretty much half of these values. But then, so to emphasize, the important point here is that you have there are differences depending on the sub area you're in. So for what might be a reasonable effect in, say, social psychology, is going to be very different to, say, if you're in neuroscience. So you're probably going to find larger effects in neuroscience if you're having lots and lots of trials. If you're focusing on like purely biological concepts, the effects are probably going to be stronger than if you're looking at things in social psychology. But you need to think about what effect, like this idea of a smallest effect size of interest. So based on my area of study, what effects are likely, and which wouldn't I want to miss out on? So we're going to get to the practical bit. Rather than you just sit here and listen to me, hopefully you've either got G-Power or Jamovi downloaded, and we will run through some examples of, for these kind of effects, essentially trying to provide us with some values if you're going to go through this process. So we're not really questioning the effect size at the moment. But we're going to go with that. When in doubt, 0.43. We're talking about this medium effect, and see where that takes us. So if you have G-Power, you'd need the t-tests and the means for two groups. And we're interested in the a priori at the moment. So based on a given effect size, how many participants would we need in order to detect it? If you're in Jamovi, you need the G-Power, the J-Power package downloaded. So in the top right, you can install different modules, and you need one called J-Power. And it will give the option to do either like an independent samples t-test, paired samples, or one sample. For this one, we need independent samples. And we're making a couple of assumptions. So it's for two-tailed. We've got an effect size of 0.43 and 80% power. So if people have that up and they want to have a go, again in the comments, how many participants do we need in each group in order to detect an effect of 0.43 between two groups? So slow down a little bit. If so, people can comment. If you have any problems or questions, please do type in the comments. So there's an aside while people are working on that. You can get like in Jamovi, it gives you the power curve by default, so it will be below the results. You can get it in Jamovi by clicking the XY plot for a range of values button. But you have to do it separately. Okay, so the comments, just to draw people in. Like Natalia, Maria, they're correct. So it's 86 in each group, 172 overall. With the others, for example, Maximus put 68. By any chance, have you forgot to put two-tailed? So potentially, so that's slightly smaller. So make sure that it's two-tailed. Make sure it's 80% power. Yep, that's the issue. Yep. And this is a nice exercise. So in one of the things I've included in materials about a year ago to help some of my students, I wrote a guide to using G-Power, which I've included in the folder for this workshop, and go through G-Power. And some of the exercises I like to do is when you're designing a study, obviously you have these decisions to make. So this is assuming you're still in the design phase. And getting people to do that. So for example, if I was to change it to one-tailed, what impact would this have on the number of people I would need? So if you have like a very strong directional hypothesis, Perhaps like a one-tailed test is going to be far more useful to you and save resources, especially if you can combine that with something like pre-registration to say I'm specifically interested in this test. It can help with sort of efficiently using resources. If you increase power, you're going to need more participants. So you can play with parameters. You can see what happens to the number of participants you need. So thank you for doing that. So moving on, we can then flip it around and say, oh, I didn't actually do a power analysis when I designed the study. Now I've got the sample size. What effect size would this reliably detect? So we're still focusing on two groups. So if you're in G-power, you need to switch between type of power analysis a priori and go to sensitivity. And for 40 participants in each group, what effect size could you reliably detect with 80% power? If you're using Jamovi, you need to switch between the option of n per group and effect size, and it will allow you to put in the group sample size. So same thing, give people 30 seconds or so. What effect size would we be able to detect with 80% power if we knew we had 40 people in each group? Yeah, this is all looking good. So almost everyone's put 0.63, so that's the answer we're looking for. There was one of 0.476. By any chance, again, is it a similar issue? Make sure you've put two tails instead of one tail. Make sure it's 80% power. So that's obviously slightly lower. So think about the factors that could mean that you're trying to detect it's saying you could detect a smaller effect size. So if power is lower, if the sample size is bigger, one tail instead of two tail, that kind of thing. So, and then for the next demonstration, we're going to switch to, rather than two groups, we're going to think about two conditions. So instead of between subjects and separating people into multiple groups, what happens if we actually want to do a within subject study and get people to do multiple conditions, try and save a little bit of time. So in G-power, we need to switch to the statistical test, and now we want the matched pairs option. So between two dependent means. If you're in Jamovi, you will need to go to the J-power drop down and go for a paired samples test, t-test instead. And we're going to start with the same thing. So for the next important demonstration, we're going to put an effect size of 0.43 in again. Two-tailed, 80% power. In Jamovi, same thing. So we're calculating n for a given effect size. Now, how many participants do we need in order to identify the same effect, but for a within subjects design? We're getting faster. Yeah, this is looking good, everyone. So for 80% power here, we need 45 participants. So this is like a big difference between if we had two groups with the same effect size, rather than 172 people in total, we need 45. So one of the things I like to emphasize to my students when we're doing this kind of thing is if resources are an issue, think about whether within subjects design is going to be feasible. So obviously not every study can be within subjects. If it's important that say the stimulus can't be seen by both groups, if you don't want sort of carryover effects or those kind of things, perhaps you just can't get away from being different groups. But if you can at least convert some factors to within subjects, it's just going to save you so many resources. So for the same effect size, it's approximately half the participants, depending on how strong the correlation is between the two conditions. With DZ, the correlation is taken into account, but if you're just working this out yourselves, the stronger the correlation, the sort of more you save in terms of the fewer number of participants. And the next thing, so say we've done a within subjects study, we know our sample size, what effect size can we detect with 80% power? So changing it to sensitivity in G-power, changing it to effect size in Jamovi. What effect size can we reliably detect with 30 participants? Awesome. Yeah, so about 0.53. So depending on how you're rounding, it's going to be about that. So we can detect with 30 participants and within subjects design around our sort of medium effect size boundary. So hopefully you can see that the difference between the number of participants you need to adequately power a within subjects design is far lower than a between subjects design. So when you're going about it yourself, think about if these are some of the things you can tweak. If you're in the design phase of a study, can you do it within subjects instead of between subjects? Can you pre-register like a one-tailed hypothesis to save you participants? Do you need more power? So rather than looking at 80% power, what happens if it's 90? Do you have enough resources to do that? So thank you everyone for taking part in that. Hopefully that was a little bit more engaging than just listening to me talk. And then the thing we're going to finish up with is, in psychology, most studies, there's very few examples where all you do is look at two groups, two conditions. We usually use slightly more complicated designs, slightly more complicated models. So this past exercise was a good demonstration for the concepts, but then for a lot of areas of research, it's going to be quite a big oversimplification, and it's not as easy to just say, right, I just expect an effect size of 0.5, see where that takes me. So we're going to see how we can then apply what we've learned to a factorial design. So for example, if you've played around with G-power, you might notice that it will try and give you power for interaction. So if you look at like F-tests, and you can see like a big drop-down menu of all these different types of tests. So it will give you some values, and I've used it in the past very badly, but it's probably wrong. So in the references, it's this blog post by a guy called Roger Gina-Sorolla. And one of the issues is it gets you to define an effect size for like the omnibus effects. So for example, when you're reporting like an ANOVA, you might report something like partial eta squared or omega squared. And that tells you the proportion of variance explained by your independent variable in your dependent variable. But when you're trying to think of this in terms of power, omnibus effect sizes like that are pretty uninformative because they're so general that they don't, for one of those values, you could get an almost unlimited different combination of means that could produce that exact effect. So for example, if you was looking at like a two-way mixed design, you need to think about what you're interested in. So is it just the main effect of your first independent variable? Are you just interested in the main effect of your second independent variable? Is it the interaction you're really interested in? So if you're just putting in one value to say, right, I expect like a partial theta squared of 0.04, each effect, and if you have pairwise comparisons, if it's got more than two levels, it's not just going to have one effect size. And because there's not one effect size, it means that there's no one value for power as well. So for example, this is a 4x2 design. So it's a study I include just analyze. It's got open data, so it's fun to play around with. So you've got a 4x2 design. If you're interested in the interaction, it's not that any one value is going to be terribly informative. So you've got roughly 4 comparisons you can make here. So we've got a comparison 1 where we're looking at the difference between 2 conditions in one level of the independent variable, 2 conditions in the second level, 2 conditions in the third, 2 conditions in the fourth. And each of these may not have one effect size you're interested in. So if you're trying to think of, am I interested in the main effect? Am I interested in the interaction? You need to think about the actual specific comparisons you're interested in making, because each of these will not have one constant value for power, because they're probably not going to have one effect size. So what we're going to work on now is how you can break it down to think about the effects you're interested in and making sure that you power the ones you want to focus on. So for example, this is where R and Python and being able to manually program this like a simulation approach to power analysis really comes in handy, because you can set all those different parameters. So you can say for this condition, we expect this difference. In this other condition, we expect another difference. But what we're trying to do is make it accessible. If you don't know R and Python, it's going to be a nightmare trying to go into it to simulate power analysis. If you are familiar with this, like the key principles are like before. So you set your parameters, you know the test you're interested in, you know the effect size, you know the power you're interested in. You run it over and over again, it's that long run frequency, and you're counting how many times you get a significant result, how many times you're not getting a significant result. And that's where power comes in. And you know, once you hit over 80% of those values being significant, you know that you have the number of participants you need in order to detect that effect, well, those parameters with that amount of power. But what we're going to do instead is, focus on these guys, Larkins and Caldwell, made like a Shiny app. So they used R, but made it like more menu-based, like G-Power, Jamovi. And you can do rough power analyses in the first one, which is what we're going to do today. But if you want slightly more specific like design, so for example, if you need to vary correlations depending on the variable, depending on the conditions, if you want to set different standard deviations depending on the conditions, if you want to say some of the assumptions could be violated, there is a second option where it's going to take a lot more time to compute, but it will allow you to do this slightly more accurately depending on these other things. So we're going to use the first one. So let me know if you can't find the link and I can copy and paste it into the comments. And what we are going to do is recreate a power analysis I did for one of my studies. So I had a 2x2, oh, thank you, Elena. I had like a 2x2 mixed design. So I was interested in, so this was the last study I did in my PhD. And I was comparing daily and non-daily smokers. And then it was on a reaction time task where I also varied whether pictures were displayed for 200 milliseconds or 500 milliseconds. So if we think about this in terms of design, I could be interested in the first main effect. So is there a difference in daily and non-daily smokers? The second main effect of, am I interested in the difference between the presentation times? And then the interaction are, for the difference between the presentation times, is it different, between daily and non-daily smokers for 200 milliseconds? And is there a difference between daily and non-daily smokers in the 500 milliseconds condition? And what I was interested in, I couldn't really care less about the presentation time effect on its own. It's pretty much meaningless. But what I was specifically interested in was that interaction. So I was interested in comparing daily and non-daily smokers in each of the presentation time conditions. So rather than say just one omnibus effect size just for one eta squared, I'm focusing more on those differences I'm trying to focus on. So if you open that up, hopefully you've got the slides downloaded because it might be a little bit small depending on your screen. I will walk through, if you want to recreate this on your screen, I will walk through the key things. So the first input we need is the design. So here by default, conveniently, it's already set to 2b times 2w. And what that is saying is we have a mixed design, 2 levels for a between subjects variable, 2 levels for a within subjects variable. You can change this. So one of the things in the power analysis guide I've linked to, I'm going to, at the moment, there is just a section saying, have a play around with this. But for a while, I've been meaning to update it. So sometime soon, over the next couple of weeks, I'm going to actually walk through the different options. But say if you wanted a third independent variable, you could add another one in. So you could put star another 2w, and it would be a freeway design. You have the option to do to enter the factor and level names to make it a little bit more clearer. So here we would. So to recreate the figure we did I showed on the previous slide, we have smoking, daily, non-daily, SOA 200 milliseconds, 500 milliseconds. And what we're doing here is defining each factor and the levels within it. So you need to do it in order. So the first word being the factor name. So here we just have smoking for the smoking group. And then the levels of that independent variable. So we've got 1 independent variable of smoking and then 2 levels of daily and non-daily. We then have the second independent variable of SOA, which is the presentation time of the task, and then 200 milliseconds, 500 milliseconds. We're going to start with a sample size per cell of 25. The standard deviation that I was assuming, so right at the start, I was talking about when you're trying to select the effect size you're interested in, the best case scenario is you have an informed understanding of like the summary statistics you're expecting. So the difference in means, the standard deviations you can expect, because once you go past different groups, different conditions, this is the kind of information you need to have at least an informed guess, because again, remember, we don't know the true effect size. We're trying to have like a conservative estimate. If anything, you want to slightly overestimate the sample you will need, because power follows a curve. If it happens to be, the effect size happens to be bigger than you thought, great. If it's smaller than you thought, then you're gonna have lower power than you anticipated. Because we have a within subjects factor, we need to suggest a correlation. So remember, the stronger that correlation, the easier it will be to find the difference between them. So we're going to assume a 0.8 correlation, the reaction times from the same participants in different conditions. So it's going to be relatively high. This is a value that you would hopefully identify from previous research you've done. If you can find open data from similar tasks, trying to estimate these values. You could always vary it as well. So you could start by, say, assuming 0.8. If you think it could be lower, what happens if you drop it to 0.5? How does that impact power? And then the final thing that we need is we need to define the mean for each cell. So this follows the order that you entered the factor and level labels. So A and B being independent variable 1, independent variable 2, and then the numbers being which levels. So for both of the daily smoking groups, we're assuming that it was like a mean of 0. So my personal approach is I like to express it relative to 0, because then the difference will essentially be expressed in like Cohen's d, in standard deviations. Because if you know there is a 5 millisecond difference between daily and non-daily in one group with a 20 millisecond standard deviation, that is really straightforward. That is really straightforward to work out as like Cohen's d, because it's just the mean difference divided by the standard deviation. So 0 and 0 for both of the daily smoking groups. And then for non-daily smokers, we have like a 5 millisecond difference in the 200 millisecond condition, 500 millisecond difference in the 10 millisecond difference in the 500 condition. So we have to define each mean that we expect for the groups. And if you click Setup Design, that will create the figure on the right. So this is your kind of sense check of have you entered all the data as you anticipate. So because we're assuming 20 seconds, a 20 millisecond standard deviation, we can see there's quite big error bars spanning plus or minus 20. For both of the daily conditions, we have it on 0. And then in our non-daily group, we have the 200 millisecond condition at 5, the 500 millisecond condition at 10. And we can see that's exactly how we wanted it. And in the design, it says the sample size is 25 per group, and that's what we're going to start with. So if you click Setup Design, should show that on the right. We now need to go to the next column, which is exact power, which will look a little bit something like this. So we set up the design in the first tab. We now calculate power for the design that we've set up. So what you should do, you should just need to click Show Results of Simulation, and you should get the results I see on the right. So please let me know if you see things slightly differently, and we can try and explore where the error is. And what it will do, so reiterating this point of you don't get one value for power. So depending on those pattern of means, you're going to get different values depending on the effects you're anticipating. So we get the power for the main effects in your ANOVA. So we get main effect of smoking, main effect of presentation time, and the main effect of the interaction. So purely by coincidence, the power is all the same for each of them, for each of the main effects. And we can see that's at about 28%. So it's pretty low. We then have, for the individual comparisons, the power as well. So this is like we're conducting your pairwise comparisons, like your t-tests on the difference in conditions. And I'm not interested in all of them. I was interested in daily and non-daily for 200 millisecond difference, daily and non-daily for the 500 millisecond condition. So you can see for the first comparison, it's about 14% power very low. And for the 500 millisecond condition, it's about 40%. So it's a little bit higher, but it's still not ideal. What I would like you to do now as the last thing before we finish is all those values of power were very low. How many participants would you need in order to reach 80% power? So what you will need to do is go back to the design, and you can adapt this. And you will have to play around with the sample size per cell. So if you try higher values, click Setup Design, go back to exact power, and show results of simulation. Those values will update. Having a little play around with it, how many participants would we need in order to reach 80% power in these comparisons? So remember, we're looking for the lowest value. So we want all of these to be at least 80% in terms of power. And if you have any problems, please do comment away. While people are working on that, one of the things I should probably say, so I rambled a little bit in the explanation, so it's about to hit an hour. I don't mind sticking around for 15 minutes or so to finish things off. If people need to go, that's up to you. It's Ilya's recording it, so you'll be able to see it later on. If you want to stick around, I'll still be here.
James, maybe while there's a pause for people to calculate all that, I have one question. I have one question. Maybe you can comment on it right now about the sensitivity analysis. So as far as I know, there is quite a substantial critics of sensitivity analysis by lichens on how useful is it actually. Can you maybe comment on that? Do you know something about that? I mean, the main idea is that is always such an overestimation of your effect sizes that maybe it's not worth doing that at all. What do you think about that?
So I hadn't seen his exact points. I didn't know he'd criticized it. So for me, it's just, it depends when you're doing it. So the thing about sensitivity power analysis, it's about, and I've only got 2 slides after this, and then we're finished anyway. And one of the points I was going to make is, what you don't want to do is, like, so you've got the a priori, so before a study, you don't want to lie to yourself. So there's no point pretending you did an a priori power analysis to inform your study when you never did one, when you designed it. So for me, a sensitivity power analysis is really helpful if you never did one in the first place, so to inform your design. So if you've already started, say, design, if you've got a study up and running and you're watching this, so that's slightly different. So I'll come on to that. So observed power is what I talked about post hoc. So observed power is useless because it's just the p-value. We're trying to think about that long run behavior. So if the idea is that if you used to repeat a process over and over, how many times would you observe different events? So with a priori, you're asking how many participants would I need in order to detect these effects with 80% power, 90% power, so whatever you're setting. On the flip side, if you never did a power analysis when you was planning and you're trying to work out how sensitive your study was, the question turns into, if I have 40 participants each of my groups and repeated this process over and over, what effect size could I detect with 80% power? So the question is slightly different. So that's going to be slightly different to observe power, excuse me. That is the same as post hoc. Sensitivity is slightly different when it's, you're asking, for me, it's calibrating what I would expect to find. So if I say I could only detect effect sizes of like a Cohen's d of 0.8, I know that I never had much of a chance to find smaller, more realistic effects. Kind of calibrating my expectations of what I could find. If I did a power analysis and say, oh, I had 80% power to detect effect sizes of 0.2, and I've covered all of the most likely effect sizes, and the ones that have much lower power to detect are effects that are that small that they may not be practically meaningful anyway. Does that answer that?
Yeah. Thanks a lot.
That's okay. So how's everyone getting on with this? So moving from, all we're doing is changing the sample size per cell from 25 to try and get those next values above 80. So I'll go back to that just in case people need to copy down the parameters. Yeah, that's good, Alexandra. That's pretty close. So wait for a couple more people to post answers. So you're looking for around what Alexandra has, and I'll tell you what I got after a couple more people have commented. One thing to maybe emphasize as well. So this is sample size per cell. So because we've got a between subjects factor, going by Alexandra's figures, that means you're looking at around 200 people in total. Right. Well, I think we'll move on from this. Like, you'll have the slides. So I essentially get between it's between 101 and 105. So 101 is like the first figure over 80. So Alexandra's almost there. You can play around with it like that. Like, so 105, I think, turns out to about 81 point something. So it's the first like round figure that gets you over 80% power. And just to reiterate, you have to think about what effects you're interested in. So if there are some comparisons you're not going to be focusing on, you don't need to worry about the power between those. We're interested in the comparisons we are specifically interested in. So there's final two things, and then we'll have some questions. Again, if people need to go, that's fine. I slightly overestimated how much we could get through in an hour. So for some closing points, comes on to what Ilya asked. If you're in the middle of a study, don't feel that you need to make up power. So you see reviewer comments about this all the time, like, oh, you need to report a power analysis, where if you never did one in the first place, you're just lying to yourself and everyone. So although it can be difficult and some reviewers will insist on it, you kind of just have to push back and go, I didn't do one. The best I can do is, now that I've finished the study, this is, you essentially report like a sensitivity power analysis. So I know I now have 100 people in my sample. This is the effects. These are the effect sizes I would be able to reliably detect. These are the effect sizes I would not have been able to reliably detect. The best case scenario is when you combine power analysis and pre-registration. So Ilya is telling me that this is the main focus about why I was invited today. Just because it's very rare still to see power analyses reported. And then you get some that are, for me, far too coincidental, where it's just like, oh, we anticipated this precise effect size that just happens to give us the amount of participants we had available. And it's like, really? So it's all about being transparency at the end of the day. So you can state in advance how many participants you plan to sample. If this is different when you finish the project, all you need to do is transparently explain why there is a difference and report if the sample size is lower than you anticipated. Again, it then flips to sensitivity. If your sample size was much lower, what effect sizes would that mean you could reliably detect? If it's higher, it's less of a worry. You just need to explain why it's higher than you planned. And for the final thing, so if you want to move on from this, I appreciate the last one maybe was a little bit of an ask in the time available. So if you need to work on this after, the slides and support materials are going to be on here. I'm going to update my power analysis guide very soon. So hopefully over the next couple of weeks, I just need to make some time to do it because all my students ask if they do a factorial design, how they can approach it. And then what I have done, so rather than have like a reference list at the end at like top size 5 font, I've created a reference list separately with an annotated bibliography at the end. So it covers things like, if you want to learn more about power analysis in general, if you want to learn more about like a simulation approach, if you want to apply power to more complicated models. So I've got quite a few resources on, say, mixed effects models and power analysis procedures to those. So Elena, yeah, so because it's based on like a Shiny app, you do get it. So is it working on mine? 1 second. Oh no, it's still working. Yeah, if you open it on just the first page, or maybe I overloaded the server, getting like 40 people to look at it. Yeah, but it can be, if you don't use it for a while, or say like a lost connection to the server and you just need to refresh it and it should work. But maybe I overloaded it. That is everything I had planned. So I hope that was useful as a starting point. No worry, Maximus. Thank you for attending. If people have questions, I don't mind sticking around 5-10 minutes. If you want to ask anything like verbally, if anyone has any questions, feel free to type away. If you need to go and you want to follow up, always welcome to email me. I moan a lot on Twitter, which is where Ilya found me. So I'm there as well. Yeah, Igor. For the one we're using, it should be okay because it's this exact version. We're not varying lots and lots of different parameters. So in the other version, it's based on a simulation approach where you can vary excuse me, you can vary, say, like the correlations between conditions. You can vary the sample size per cell. You can vary whether, say, the like the standard deviation between different cells. So then it becomes much longer. And if you have, say, more complicated models where, say, if you're trying to calculate power for a mixed effects model, then it starts to take like minutes, hours, depending on how complicated you're getting. If you're using the exact one, it should be pretty instant. I think we just overloaded the server. But yeah, if participants gets quite high, parameters turn a lot. If you're having lots and lots of simulations, then it's going to take longer and longer. But no, thank you.
So are there any questions, guys? There are some.
Yeah, so Maria, yeah, so if I can make something up dead quick. This is going to be absolutely meaningless, but just to... Oh dear, there we go. Right, now we'll forget that rather than just me making things up. But to go to, who is it? Yeah, Maria. So if you looking at lots of different, no, sorry, Dimitri, interaction between two of subjects factors. Yes, you can do that. If you have, say, so if you just got a two-way design, it's going to be very simple. Rather than we defined 2b by 2w, it would just be 2w by 2w. And that would look, you could be able to find the interaction there. Say if you're doing like a three-way design where you've got one between subjects factor, two within subjects factors, and you're interested in the interaction just between two of those. If you go to that second page, so if you're on, say, that page, rather than the like three factors here and then these comparisons, you get the whole list. So it would be like 6 effects if you've got a three-way design, and then you would have however many pairwise comparisons you have, and it will break it down by that number. Go through some of these questions. Maria, so correlational design. So you can do correlations in G-Power. You can, I can't remember the exact heading off the top of my head. If you go to the guide array, I've got correlations in there. As in comparing patterns of correlations for several groups, do you mean comparing, say, a correlation in one group to the correlation in another? If you can just clarify that bit, I'll get back to that. Oh, yes, very quick. Yeah, so there is a particular type of test that you have to do to compare them. I think it's a Z test, so it's like a meta-analysis.
Called Fisher Z test, I think.
Yeah. So I think you can do that in G-Power. So it's going to be very similar, and I believe you can do it. I can follow it up if you want to play around. If you've got any questions, I can look into it. But I'm pretty sure you can. I'm pretty sure it's under, if you go on types of test or family of test, it's under exact, if my memory serves me correct. Yes, perfect. How do you recommend to read about power analysis and mediation? I had one, I'll remember, there is one study I had, like, so personally, I do not know much about mediation. I will be happily admit, all my research is experimental, so I've never focused much on mediation models. You can do it. Again, it's just an extension of this. So you have to define your parameters, repeat it over and over, how many times you observe different results. You can't do it with the things I've introduced today. So that is where simulation approaches are going to be the thing you have to do. I did come across one study on power analysis for mediation. I will update the reading list with that in. So if you check back, I will add a study into there. And yes, Elena, so post hoc power, absolutely useless. It's just for p-value. You can just call the p-value post hoc power. That's all it is. And that's my personal approach. So if I say work on a study that I either whoever planned it didn't do a power analysis at the start, or if I have a study that I didn't do a power analysis from the start, then the sensitivity power analysis is the next best thing. So because it's, for me, it's like calibrating expectations. So based on the total sample size I end up with, I know what I was likely and unlikely to find, given like an effect size. So you can think about if you observed like significant results, you can think about what you was likely to find, and then it turns back onto what are your like meaningful effect sizes. So for your smallest effect size of interest, is that contained within it? So could you reliably detect that effect size? Cool. Any last questions? So more than happy for people to get in contact with me afterwards. So I mainly use it in experimental designs. The best I can do is point you to some resources for things like mediation that I've not done directly, but I can probably find something to help. But if not, thank you very much for having me. It's been fun.